Okay, so we've covered nucleic acids, DNA and RNA, we've covered chromosomes, and now I want to talk about gene expression. So first of all, what is meant by a gene? Well, a gene is a unit of DNA that contains the instructions to build a protein. And why is this important? Why are genes important? Well, proteins are the molecules that are going to build and maintain the body. Antibodies, enzymes, skeletal muscles, all of those are made out of proteins. And so genes are important because those contain the instructions to build those proteins and to continue building them throughout life, and so the body can function. What does gene expression refer to? Well, gene expression refers to taking a gene and actually turning it into a protein, taking those instructions and going through the process to actually make a protein. Let's take a look at this figure down below. Here we have a chromosome, and as we learned earlier, we have DNA that is packaged within these chromosomes. And the DNA is divided into different genes. So this section of the DNA represents gene 1, and this section of the DNA represents gene 2. And now they're showing us the specifics of gene number 2. And what we're looking at here is the actual gene sequence. All of this lettering that we see represents the actual gene sequence. So the question is, which part of a nucleotide, we know that nucleotides make up DNA, which part of a nucleotide makes up the actual gene sequence, the actual instructions that we use to make a protein? So what's being represented by these letters here? What are nucleotides made up of? They're made up of sugars, deoxyribose sugars. They're made up of phosphate groups, and they're made up of bases, nitrogenous bases. And which one is being represented by all of this lettering? Those are the nitrogenous bases. And so it's the order of these bases that's actually represents the gene sequence, that actually represents the instructions to make a protein. Now here we have the definition of a gene. A gene is a unit of DNA that contains the instructions to build a protein. Genes code for proteins. And there are about 21,000 genes in the human body that code for proteins. There are about 21,000 what we call protein coding genes. And this complete set of genes is referred to as an organism's genome. So there's 21,000 protein coding genes in the human body, and all of those genes are collectively referred to as the genome. So the question is, where is the smallest unit that we can find an organism's genome? Can we find it on each chromosome, on each homologous pair of chromosomes, dispersed between the chromosomes in each cell, or dispersed between chromosomes in different cells? And the answer to that is C. We can find an organism's entire genome dispersed between the chromosomes in each individual cell of the body. And we can go back and partially visualize that. Here we are looking at a karyotype. And again, this is a photographic representation of all of the chromosomes within a cell. And we're looking at a somatic cell here that has 46 different chromosomes in it. And those 21,000 protein encoding genes are dispersed between these 46 chromosomes. And just remember that each of these is a homologous pair, homologous chromosomes, and the chromosomes within a homologous pair contain the same genes, but they don't contain all of the genes. Maybe homologous pair 3 here contains 1,000 protein coding genes. Maybe homologous pair 4 contains 3,000 protein encoding genes. But collectively, all 21,000 of those protein encoding genes are dispersed between these 46 chromosomes within each cell of the body. Next question. The genes present in liver cells, what we call hepatic cells, are the same as the genes present in skin cells. Is that true or false? That's true. We find the same 21,000 protein encoding genes within each liver cell, and we find those same 21,000 protein encoding genes within each skin cell. Let's take that a little bit further. The genes expressed, the key word here is expressed, the genes expressed in liver cells are the same as the genes expressed in skin cells. Is that true or false? Now the answer to that is false, because even though skin cells and liver cells contain the same genes, they don't express those, they don't express the same genes. Maybe the liver cells only express 5,000 of those genes, whereas the skin cells only express, maybe 2,000 of those genes. And further, these 5,000 genes that the liver cells express may be different, or at least partially, than the 2,000 genes that the skin cells express. And that's the reason why liver cells both look different and function differently than skin cells. That's why different cells look different. That's why different cells look different from each other, and why they function differently is because out of those 21,000 genes, they express only a certain set of those. Two steps are required for gene expression, and those 2 steps are transcription and translation. In general, what's happening with each of these steps and where does this occur within the cell? So here we have a cell of the body, and we're looking at the nucleus. Here's the DNA within the nucleus. And the first thing that we need to do to be able to express a protein, to make a protein, is we take that DNA, and specifically we take the gene that we want to express, and we make a copy of it. And the copy of that gene that we want to make is called mRNA. We make a messenger RNA copy of the gene that we want to express. And that step, taking a gene and making an mRNA copy of that, is called transcription. And that occurs within the nucleus of the cell. Then what happens is that mRNA moves out of the nucleus into the cytoplasm. And now what we do is we take that mRNA molecule, we read those instructions on the mRNA molecule, and we make a protein from it. And that's what this is representing. That's a string of amino acids, which officially is called a polypeptide because it's still in its primary linear structure, but eventually it'll go to its 3-dimensional structure and be called a protein. But to get back to the point, the mRNA travels into the cytoplasm, and that mRNA, the instructions of that mRNA are read, and we make a polypeptide from it. And that step, taking an mRNA and actually making a polypeptide, making a protein, is called translation. And that occurs within the cytoplasm of the cell. Okay, DNA is a double-stranded molecule, yet only one of these strands is used to make mRNA. what strand is used to synthesize mRNA, and then label that on the diagram. So here, oops. Here is our double-stranded DNA. And we can label both of those strands. One of those strands is called the coding strand, and the other is called the template strand. And only one of these strands is going to be used to make an mRNA molecule. And which one is that? Here you can see now we're looking at transcription, and we're making an RNA molecule, an mRNA molecule, based off one of those strands. And which strand are we using? We're using the template strand. Okay, now we're looking at some of the specifics of transcription, taking that template strand and making an mRNA molecule from it. And first what I want to do is label some things. RNA polymerase, the template strand, mRNA, and free RNA nucleotides. Let's do some labeling. First of all, where is the RNA polymerase? Well, this big purple blob is the RNA polymerase. Now let's look at the DNA. There's the coding strand, which makes that the template strand. Where is our mRNA molecule? This is our mRNA molecule that we're making. And to make mRNA is nucleic acid. It's made of nucleotides, and so we need building blocks. We need free nucleotides to make that mRNA molecule. And where are those represented? Those are our free nucleotides. So all of those things that we just labeled, RNA polymerase, the DNA template strand, mRNA, and free RNA nucleotides, these are all the components that are required for transcription to occur. And so let's take a little more, get into a little more detail. And specifically, I want you guys to look at this figure. This is a similar figure from above. And I want you to tell me what RNA polymerase is specifically doing. Now, what type of molecule is RNA polymerase? Ase, the suffix, means that it's an enzyme. And what type of enzyme is it? Or what is it going to do? It's going to make polymers of RNA. So RNA polymerase is an enzyme that's going to make the mRNA molecule. And the question is, how does it do that? Well, the first thing that the RNA polymerase has to do is it has to take this double-stranded DNA and it has to unwind it. So the RNA polymerase unwinds the double-stranded DNA. And the reason this is important is because now that gives the RNA polymerase access to this template strand of DNA. What happens next? Well, the RNA polymerase is moving along the double-stranded DNA. And what happens is that as it moves through the double-stranded DNA, it reads the nucleotides of this template DNA strand. And when it reads a C, what does it do? When it reads a G, what does it do? It adds a C onto the mRNA molecule. When it sees a T, what does it do? It adds an A onto the mRNA molecule. And so it's reading this template strand and it's adding complementary bases to the mRNA molecule. It moves along, it sees this T, it adds another A. Here's where it gets a little bit, not tricky, but something you have to be aware of. What happens when the RNA polymerase gets to an A nucleotide, an adenine nucleotide on the template strand? Well, it's going to add a complementary base. And what's complementary to A? Get my pen working here. There we go. T is complementary to A. But do we find thymines in RNA molecules? We don't. Instead of thymine, what do we find? We find uracils. So when the RNA polymerase comes upon an A nucleotide in the template strand, it adds a U, a uracil, on the RNA molecule. All right. To begin transcription, RNA polymerase binds to a location on the DNA molecule called what, which is upstream of the region to be transcribed. So down on this figure, this represents the gene, and the RNA polymerase it's not just going to bind initially right in the middle of the gene, it binds to a region upstream of that. And this is the region specifically that RNA polymerase is going to bind to. And what do we call that region? That's called the promoter region. So it binds to the promoter, and then it's going to make its way down the double-stranded DNA, unwind that DNA, and make an mRNA molecule based off of the template strand. So what we've done is we've taken this template DNA strand and we've made an mRNA molecule from it. And this mRNA molecule contains the instructions to build proteins. Specifically, this mRNA molecule contains the instructions to add specific amino acids onto the protein that we want to build. And so the question is, what is the unit of mRNA that contains the instructions for adding a specific amino acid onto the polypeptide chain? And the answer to that are these guys here, which are groups of 3 mRNA nucleotides that we refer to as codons. So a codon is a group of 3 mRNA nucleotides that contains the instructions to add a specific amino acid onto the polypeptide chain. Each one of these 3 sets of nucleotides represents a codon. And based on each of these codons, we can actually determine what amino acid is going to be added onto the chain. Let's take a look at the first one, U, U, U. What amino acid is going to be added onto the chain when we have a UUU mRNA codon? And to determine that, we use this figure. And you notice that all of these represent the different 20 amino acids that can build the body. And so the way we determine what codon represents what amino acid is, first of all, the first letter is a U. So here we have the first letter, and here we have a U. Then what's the second letter? The second letter is also U. And so you go over here, here's the second letter, and then the third letter is also a U. So that takes us right there. And the amino acid that's going to be added with a UUU mRNA codon is phenylalanine. Let's look at, so this is going to be phenylalanine. Let's look at the second one, GGC. First letter, G. Second letter, G. So we go over here to the second letter. Now we're dealing in this box. And the third letter is C. There we go. The amino acid glycine is going to be added for the GGC. And on the exam, I'll give you this chart here. You don't have to memorize this chart or anything, but you do have to be able to, you have to know how to use it. I'm going to give you a sequence of an mRNA molecule. You're going to have to identify individual codons, and then you're going to have to use those codons to determine the amino acids that are going to be used to build the protein. All right, below is a partial DNA sequence for the COL1A1 gene. And this provides the instructions for building a type of fiber called collagen fiber. Collagen fibers are super tough. We find them in tendons. We find them in ligaments, places that we need to really resist stretch. They have a really high tensile strength. And specifically what I've given you is the coding strand for that particular gene. And what you guys need to be able to do is, based on that coding strand, you need to give me the template strand. Based on the template strand, you need to be able to give me the mRNA sequence. And then based on that mRNA sequence, you have to give me the polypeptide sequence, the sequence of amino acids that are going to be added on. So let's go through this. First of all, let's complete the template strand. How are we going to do that? Well, the key here is complementary base pairing. The double-stranded DNA molecule is held together by complementary base pairs. So I'll just complete a partial sequence of it here. What is the template strand going to be for this gene? C, T, G, T, A, C, A, G, T, A, T, T, C. So you just made yourself the template strand. And that's easy to do because you know that these are complementary to each other. Now we need to make an mRNA strand. So we're going to use the template strand to build that through a process called transcription. And what is the mRNA strand going to be? G, A, C, A. Now what's complementary to the A here? That's going to be U, G, U, C, A, U, A, G. And each one of these is an individual codon. Now, out of this mRNA sequence, where's the start codon? And the start codon is where translation is going to begin. What's the start codon here? The start codon is AUG. So we're going to translate the start codon and all of the codons that are downstream of that. And then lastly, let's just give the resulting amino acid sequence. And we're going to start with the start codon, AUG. A, U, G. There we go, methionine. Then we have UUU. Here's our UUU, that's our phenylalanine. I'll just CAU, CAU, that's histidine. And then lastly, held together by covalent bonds, peptide bonds. And lastly, AAG, lysine. Now let's look at some of the specifics of translation. And again, what we're doing with translation is we're taking an mRNA molecule and we're using those instructions on the mRNA molecule to build an actual protein. And that occurs within the cytoplasm of the cell. And in this figure here, we have all of the necessary components that we need for translation to occur. So let's just go ahead and label some of these. First off, what is that molecule? That's our mRNA molecule that actually contains the instructions to build the protein. What about this long string right there? That is our growing polypeptide chain. So as the instructions for mRNA are being read, we're adding individual amino acids onto this polypeptide chain. We're actually building a protein, and that represents the protein that we're building. What about this guy? That is called the ribosome. And the ribosome represents the molecular workbench where translation is going to occur. So to build this protein, we need some structure that this is going to occur on and occur within. And the ribosome is the molecule where translation is going to occur. What are these guys out there floating within the cytoplasm? Those are tRNA molecules, transfer RNA molecules. And attached to each of those tRNA molecules is a specific amino acid. And let's see, lastly, within the ribosome, we have these 3 sites, and this is called the E site, that's called the P site, and that's called the A site. Okay. So how does this process work? How does translation work? Well, what's going to happen is this mRNA molecule is going to thread itself, it's going to move itself through the ribosome. And what you can notice here is that in each of the sites, the EP and A site, there's a codon. mRNA codons are located there. And what's going to happen is in the A site, we're going to have tRNA molecules with amino acids attached to them. Those tRNA molecules are going to bind themselves to this codon in the A site. Now the question is, which tRNA molecule is specifically going to bind to the codon in the A site? Well, tRNA molecules, on one end they have an amino acid attached to it. On another end, they have what's called an anticodon. And the specific tRNA that binds to the specific mRNA codon is going to have an anticodon that's complementary to the codon. So the codon that's being read is GGC. Which tRNA is going to bind? The tRNA that has a complementary anticodon. And what's complementary to GGC? CCG. And so that tRNA binds to the codon, and then what happens is this growing polypeptide chain is moved over on top of this amino acid. And so what we just did is we took this polypeptide chain that we're growing and we added another amino acid onto it. Then what happens is this tRNA in the A site moves to the P site, the tRNA in the P site moves to the E site, and the tRNA that's in the E site, which now is empty because it just contributed its amino acid, is ejected. That's the exit site, and it goes back out into the cytoplasm and binds its specific amino acid. And that happens over and over and over until you get to the end of the mRNA molecule, till you get to what's called the stop codon. And then what happens is the mRNA shifts itself through the ribosome. And so now that codon is in the A site. And the process repeats itself. A tRNA that's complementary to that codon is going to bind, and then the string of amino acids moves on top of that. So we just added another amino acid to the growing chain. And again, the mRNA keeps moving through the ribosome, and amino acids are kept being added on individually until you get to the stop codon on the mRNA molecule. And for a process like this, I can kind of explain it on this figure, but it's also really helpful to look at some videos, and there's a lot of good YouTube videos out there, and I have some of those YouTube videos in the lecture material for gene expression. All right, what we're looking at here is an actual tRNA molecule. And this is an RNA molecule. And RNA is single-stranded. And so here you can see this single strand of RNA. But what happens is that it bends back on itself to form a secondary and a tertiary structure, a three-dimensional structure. And how does it bend back on itself? Well, it forms these complementary base pairs between the tRNA molecule, and that gives it structure and allows it to do what it's going to do. Here's the part of the tRNA that's going to bind the amino acid. And down below is the anticodon region. And the anticodon that we're looking at is AAG. So I want to know two things. One, what's the mRNA codon that this tRNA is going to attach to in the ribosome? And two, what amino acid is going to be attached to this specific tRNA? Let's start with the mRNA codon. What's the mRNA codon that this anticodon is going to bind to? It's complementary, so it's going to be UUC. Next question, what amino acid is going to be attached to this tRNA? Well, remember that it's the mRNA codon that contains the instructions to add a specific amino acid. So we're going to use the mRNA codon and go to that codon chart to figure out what amino acid is going to be there. And if we do that, we have UUC. So the specific amino acid is going to be phenylalanine. And a really good example of when we see gene expression at work is when we talk about the actions of steroid hormones, the mechanisms that steroid hormones have on their target cells. So first off, what are some examples of steroid hormones? Testosterone, estrogen, progesterone, aldosterone. These are all examples of steroid hormones. And steroid hormones are produced by endocrine cells. They're produced by cells of the body. And then those steroids travel through the blood to reach distant target cells. So here we have a steroid hormone. And the steroid hormone is being escorted through the blood by this carrier protein. And what happens is that when the steroid hormone gets to its specific target cell, it's dropped off by the carrier protein. Now, what do you know about the chemical characteristics of steroid hormones? Are they lipophilic or lipophobic? Well, steroid hormones are a classification of lipids. So steroid hormones are lipophilic. They like lipids. And what that means is that when the steroid hormone gets to the target cell, it can diffuse by simple diffusion straight through the plasma membrane. So now we have a steroid hormone that's in the cytoplasm of the cell. What happens next is that that steroid hormone makes its way inside the nucleus and it binds to a receptor in the nucleus. So now you have this steroid hormone receptor complex. The next thing that does is that hormone receptor complex binds to another receptor that's located on the DNA itself. And when that steroid hormone binds to the DNA itself, what it does is it stimulates gene expression. When the hormone binds to the receptor, it stimulates transcription and translation of a specific gene that's located on the DNA. And that's how these steroid hormones work. They work on cells, they cause different effects on cells by causing gene expression within that cell. So for instance, testosterone is a predominantly male hormone, steroid hormone, and some of the things that it does is, testosterone can work on the testes and it can stimulate the production of sperm. So how does it do that? Well, that testosterone acts on the testes, it goes within the cells of the testes, and it stimulates the production of a protein that's going to stimulate sperm production. So this new protein that's formed is going to stimulate the production of sperm, what we call spermatogenesis. But the key takeaway points here: 1, steroid hormones are lipophilic, they go straight through the cell. 2, they bind to receptors in the nucleus. 3, those steroid hormone receptor complexes bind to a specific gene on the DNA, and that causes gene expression of that specific gene. We produce a new protein, and that new protein is going to have whatever effect on the cell that steroid hormone has. So the protein that's produced is going to carry out the actual effect that the steroid wants to have on that target cell. Here's another good application of gene expression. Looking at this figure, first off, these are your calf muscles, your gastrocnemius medial head and lateral head that we'll talk about in anatomy physiology. And skeletal muscles are attached to bones by tendons. And the calf muscles are attached to your heel bone, your calcaneus, by what's called the calcaneal or your Achilles tendon. And tendons are made up of a tissue called dense, regular, connective tissue. And this is a drawing of that. And what dense, regular, connective tissue has a lot of is it has a lot of collagen fibers. These are big, relatively thick fibers that we find in the body, these collagen fibers. And collagen fibers are super tough. Okay, they resist stretch. They have what we call a high tensile strength. And what's going to happen is when your calf muscles contract, when they shorten, they're going to pull on the tendon. The tendon's going to pull on the bone that it's attached to, and that bone is going to move accordingly. So when your calf muscles contract, your heel lifts up, and that's a motion that we call plantar flexion. Okay, calf raises. When you do calf raises, then you're using your calf muscles. But not only do we find collagen fibers within tendons, but we find these cells called fibroblasts. So we have collagen fibers and fibroblasts. All right, so here we have a scenario. Amy is a 23-year-old female who tore part of her Achilles tendon playing soccer. And so this Achilles tendon stretched, and it stretched these collagen fibers, and it may actually have torn some. Okay, so we have some torn collagen fibers. And the question is, how is the tendon going to heal itself? How are these injured collagen fibers going to be replaced? And something I should point out is that these collagen fibers are made of proteins. These are protein fibers. So again, how are we going to replace these protein fibers? And I'll give you a hint. It has to do with these fibroblasts. So what do you think is going to happen? Well, what's going to happen, keep in mind that these fibroblasts contain the entire genome within there. Each of these fibroblasts has 46 chromosomes. And somewhere within those 46 chromosomes, there is the gene to build collagen fibers. So what's going to happen is when these collagen fibers are injured and they need to be replaced, these fibroblasts are going to undergo gene expression. And they're going to take the collagen gene, they're going to make a collagen mRNA, and then they're going to turn that mRNA into an actual collagen protein. So it's these fibroblasts that are going to produce new collagen fibers. Then what do they do with them? Do those collagen fibers stay within the cell? They don't. They're exported from the cell, and then those collagen fibers are used to replace the damaged collagen fibers. And that's how we produce new tendon tissue, new dense regular connective tissue, is gene expression of those specific collagen proteins. And the last thing I want to talk about with respect to gene expression are transgenic or genetically modified organisms. So what is a transgenic organism? What is a genetically modified organism? Well, it's an organism that has had DNA from another organism inserted into its genome. And the example I want to use for this is a transgenic bacteria that has had the human insulin gene inserted into it. So first of all, what is insulin? Well, insulin is a hormone that's produced by the pancreas, and it binds the target cells, and it allows the cells of the body to take up glucose into the cell. So the cells can then use it for cellular respiration or whatever else they're going to use it for. Now, what is type 1 diabetes? If you're a type 1 diabetic, what's the problem? Well, type 1 diabetes is an autoimmune disease where basically the body's own immune system attacks the cells in the pancreas that produce insulin. And so type 1 diabetics are not able to produce insulin. And so what type 1 diabetics need to do is they need to monitor their blood glucose, and when their blood sugar gets high after a meal, they need to inject insulin, and that allows the cells to take up glucose, and then your blood sugar goes back down. So where are type 1 diabetics going to get this purified insulin that they're going to use for daily injections? Well, that's where we use this transgenic bacteria. So here we have a bacteria, and this plasmid represents the genetic material of the bacteria. It's a circular chromosome. And what we're going to do is we're going to take an insulin gene from a human cell, and we're going to insert that within the bacterial chromosome. So now we have a bacteria that has the human insulin gene as part of its chromosome. And guess what? That bacteria is going to express the insulin gene. So what we do next is we put that bacteria in a nice environment with all the nutrients and everything that it needs, and those bacterial cells produce insulin. They produce a whole bunch of insulin. And then what we're able to do is we're able to extract the insulin that's being produced by those cells, extract it from the culture medium, and purify it. So now we have purified human insulin. And it's this purified human insulin that's produced by bacteria that is the insulin that type 1 diabetics are going to use from their injections. And I want to emphasize that this insulin that's being produced by the bacteria is the exact same, it is human insulin. It's the exact same structure as human insulin. And so why do we even use bacteria to do this? Well, when we grow bacteria in culture, they're going to replicate rapidly. And so we can have thousands or millions of bacteria, and they're all producing insulin. And so it's a really nice way to produce large amounts of insulin, which we can then purify and use for medication purposes. And the last question I want to address is, how is it that DNA from an organism can be inserted into any other organism, and that receiving organism, the now transgenic organism, will express that inserted gene? How is it that we can take a human insulin gene, insert it into bacteria, and that bacteria will express that gene and actually produce insulin? And the answer to that, the reason we can do that, is because the structure of DNA is the same between all living organisms. In all living organisms, DNA is double-stranded, and it's made up of individual nucleotides: adenine, thymine, guanine, and cytosine. Now, the sequence of the genes, gene sequences, are different between different organisms, but the structure of the DNA itself is the same. So when that insulin gene is human insulin gene is inserted into the bacteria, that bacteria just treats it as its own gene because the structure of the DNA is the same. And the RNA polymerase and all the translation machinery treats it as its own. It's something pretty amazing that all living things have in common.